Hello boys, welcome back to another session of video tutorials, and this session is meant for class 11th biology students, and this is for 5th February 2021. As we are into the revision mode, and we are revising this time around in the form of a test paper 2. Last time around we did in the form of test paper 1. Now this round we are going to do in the form of test paper 2, and the chapter that we are going to deal with is neural control and coordination. And this is important chapter not only for the sake of examination but for the general life also. We need to know what are the various functions of the nerves that are running through our body, and what are the functions of brain, how brain is the center of control and command. All that we need to understand in general day-to-day life also. Then this way of dealing revision will make you familiarized with 1 mark questions from this chapter, 2 mark questions from this chapter, 3 marks, as well as long answer type carrying 5 marks. So you understand through paper, test paper 1 and 2, what are the various areas to be focused in this chapter, how to present your answer, what are the diagrams that are very important from this chapter, label the diagram so that you understand the significance of each label. Then you are in a better place. And remember always, you must support 3 marks and 5 mark questions always with a diagram, labeled diagram. So keeping that in mind, question number 1. Name the band of nerve fibers that joins the two cerebral hemispheres in mammals. Question 2. What is threshold stimulus for a nerve cell? What is a compound eye? Eyes, there are two types, simple eye and compound eye. Next, give parts of neuron. Look at that, just now I said diagram based question. You need to label the diagram and understand the significance of each part of the neuron. Fifth, describe the role and location of ciliary body in human eye. Next, what is mosaic vision? Question 7. Why do giant squids have very thick nerve fibers? Question number 8. Where are synaptic vesicles found? Name their chemical contents. What is the function of these contents? Next, long answer type. What is meant by resting membrane potential of a neuron? How do ion channels and sodium potassium pumps contribute to resting potential? I have been telling you in the normal video classes time plus revision time, sodium potassium pump plays a very crucial role in our health, in maintenance of our health, particularly in controlling the heartbeat. If sodium increases the heartbeat, potassium decreases or slows down the heartbeat. Thereby, when both the elements, sodium and potassium, are in a balanced state, our heartbeat also will be under control. Neither it is too fast nor it is too slow. So this is how sodium potassium plays a very crucial role in the functioning of the heart. Now take a paper, start writing answers. After 30 to 40 minutes, come to the answer session. Now in the answer session, compare your answers with the answers that I am providing with. Now first question, corpus callosum. This joins the two cerebral hemispheres. Corpus callosum. Question 2. The minimum intensity or strength of a stimulus required to initiate depolarization of a neuron is called threshold stimulus. And remember, when we are talking of this depolarization, we talked about 3: depolarization, polarization, repolarization. Remember? So these are the 3 things that you must remember. Then, question 3. In insects, the eye is composed of many independent visual elements called metida. Such an eye is called compound eye. Okay, whereas we have got a simple eye. So compound eye has got many independent visual elements. So frame by frame will make the total vision, and that will be helped by ommatidia. And question 4, diagram based question. Neuron is a microscopic structure made up of three parts. One is the head portion, another is the tail portion. So head portion is called cyton, cell body, and tail portion is called axon. Okay. Now the cell body, that is this head portion, contains cytoplasm. We call it we call it neuroplasm, because it is involving neuroplasm instead of cytoplasm. You can call it as neuroplasm, with the typical cell organelles and some granular bodies called Nissl granules. So you can see Nissl's granules, and then there are branches around this cyton. Those branches are called dendrites. And you can also observe the longest dendrite makes an axon. Look at this. This is a dendrite, this is a dendrite, this so as this, and this is a elongated one. So what is axon? Elongated dendrite is an axon, right? So dendrites are short fibers that branch repeatedly and project out of the cell body. They transmit impulse towards the cell body or cyton. Next, we go with the, of course, in labeling part, you name it as cell body or cyton and nucleus. And what is most important is the presence of chemicals called neurotransmitters. Like I said, acetyl-CoA is one of the neurotransmitters. So within this neuroplasm, you got those chemicals which are very, very crucial for our stimulus. They are called neurotransmitters. And then this extended dendrite, which is called axon, sometimes it has got a covering that's called myelinated sheath. You can see if myelinated sheath is covering the axon, such a neuron is called myelinated neuron. On the other hand, some neurons, they will not have this cover. Myelinated sheath will not be there. So they are called non-myelinated neurons. And within this myelinated cover, you can see there is Schwann cell, okay, presence of a cell. And when the myelinated sheath is there, you can see the grooves here, ridges. So these ridges are called nodes of Ranvier. And of course, the tail portion, axon portion, starts bifurcating and forms synaptic knob. And the synaptic knob, with a minute gap called synapse, continue with the next adjacent neuron. So synapse is basically a small minute gap that is present between two adjacent neurons, right? Now, axon is a long fiber. That's what I said. Extended dendrite is an axon. Its distal end is branched. Each branch terminates into bulb-like structures called synaptic knob, as you are seeing this one, synaptic knob, right? Going ahead, question number 5, answer location. The ciliary body is a part of the eye that includes the ciliary muscle, which controls the shape of the lens, and ciliary epithelium, which produces aqueous humor, that liquid part. The ciliary body is part of the uvea, the layer of tissue that delivers oxygen. Look at that, this is very important, and nutrients to the eye tissue. Ciliary body also contains ciliary muscles. This muscle changes the shape of the lens when your eye focus on something. So you can imagine without a ciliary body, there will be no change in the shape of the lens. That would have made us very difficult to see the things. So thanks to ciliary body. Question 6. Uh, this type of vision is found in insects due to a presence of compound eye. A complete image of the object as seen by the compound eye is formed by number of small lineages, I said frame by frame, each of which is contributed by a metadia. Such an image formed by many bits of images is called mosaic viewing or mosaic image, and the vision as mosaic vision. Question number 7. The velocity of a nerve impulse in a nerve fiber depends on two factors. One, on its myelinated covering, just now I said myelinated sheath, and also on the thickness of the fibers. Thickness of the fibers. The impulses travel faster in thick nerve fibers. Since giant squids are very large sized aquatic animals, they have thick nerve fibers. So impulses will be very fast. Question number 8. Synaptic vesicles are found in the bulb-like expansions called synaptic knob and the nerve terminals, just now we have shown. Each synaptic vesicle contains as many as 10,000 molecules. Look at that, 10,000 molecules of neurotransmitters like acetyl-CoA acid. Substance called acetylcholine that's responsible for transmission of nerve impulses across the synapse. That is the importance of acetylcholine. When a wave of depolarization reaches the presynaptic neuron membrane, the voltage gained, calcium channels concentrated and the synapse open. Ca2 plus ions diffuse into terminal form of surrounding fluid. The Ca2 plus ions stimulate the synaptic vesicles to move to the terminal membrane, fuse with it, and then rupture by exocytosis into the cleft. This neurotransmitter diffuses across the synapse and stimulates the membranes of the next neuron. So this is how they activate adjacent neurons and the impulses continuously run through. Question number 9. It's a 5-mark question. Remember, resting membrane potential. The electrical potential difference across the membrane of a resting neuron is called resting membrane potential. The membrane is polarized, first polarization, with a negative interior, as you are seeing. Look at that, negative interior and positively charged exterior. The permeability of membrane to K plus ions, you can see inside, is greater than its permeability to sodium ions. The negatively charged protein molecules can cross the membrane. The sodium pump transports, this is important, you must remember, 3 sodium ions to the exterior, while in exchange only 2 K plus ions comes inside. Hence, you can see here, in the diagram it's shown interior and exterior. Hence, the surface carries a positive charge, you can see, which the interior is negatively charged. You can see the positive one and the negative one interior. And remember, how much is allowed? Only 3 sodium ions exterior and 2 K plus comes inside, interior. So that's how the polarization takes place. And then of course, next stage is depolarization. After depolarization, there will be flow of ions again. So diagrammatic representation of impulse conduction through an axon, that is point A and point B. So always, as I said, you must substantiate your answer with a diagram, right? Next time onwards, we will take up one more paper. That is, we have got left with one more chapter, 22nd chapter.